Okay. We are now streaming live on YouTube.
Oh boy. This is the time when the dog will start barking or a child will burst in.
Right.
Oh, I'm going to turn off my own ringer. That's another thing I always forget.
Oh, good call. All right, it's 4 o'clock, so I'm going to start the seminar by introducing our seminar speaker today. I'm so pleased to welcome Dr. Amy Angert from the University of British Columbia. She is a professor of botany and zoology within the Biodiversity Research Center at UBC. She's also a Tier 2 Canada Research Chair in Conservation Ecology. Her lab group works at the interface of ecology and evolution to understand the evolutionary ecology of species' geographic distributions, asking what limits adaptation at the edge of species ranges, and why closely related species vary by orders of magnitude in range sizes, and how these ranges are likely to shift in response to rapid climate change. And I'm so pleased to welcome Amy because these are exactly the concepts I've been teaching in Bi 370 for the past 3 weeks as we've been kicking off the term. And so her timing, the invitation was not coincidental. It aligned well with my course, but I'm so happy to have her share her research with the rest of you here in the department. Amy is available to meet with faculty or graduate students who are interested either tomorrow or Friday of this week or later in the term if that works for you. So after seminar, I'll send an email with her available times and you can connect with Amy if you'd like to follow up after this seminar. All right, Amy, take it away.
Awesome. Well, thanks, Sarah. Thanks for the invitation. And yeah, I'm excited to share with you guys some of the work that my lab group has been doing over the past few years. And in this case, it's going to be about the topic of local adaptation and how it varies over space and time. Like Sarah said, in my lab, we spend a lot of time thinking about these blobs on the maps that we know as geographic ranges. So, every species on the planet has a geographic distribution, but yet there are so many fundamental questions that remain about their ecological and their evolutionary dynamics. For example, are those boundaries of the geographic range, like I've colored here in orange, are those simply transient fronts limited by dispersal across the landscape, or are they boundaries defined by fitness limits? And if they are fitness limits, if a species has adapted across the heterogeneous environments that span its range, why would adaptation suddenly fail all of a sudden at a range margin? And so in this way, I use range limits as sort of a model system for testing really fundamental questions about limits to adaptation. What are the constraints on niche evolution, and why do we sometimes see niche conservatism? And although every species has a geographic range, species vary by many orders of magnitude in their range sizes, from sort of narrowly endemic things that only occur in a few spots in the world to species that are very widely distributed and maybe even invasive on other continents if they have a chance to get there. And so what is it about the constraints on range expansion that differ so fundamentally between narrowly and widely distributed species? And I freely admit that I would be so delighted to sort of answer these fairly esoteric questions about limits to adaptation for my career. But what's happened more and more often lately is that every empirical data set I've tried to work with to answer these questions about sort of equilibrial evolutionary processes has an imprint of climate change in it. So increasingly, my questions are, I'm sort of shifting my focus to ask not about evolutionary and equilibrial ranges, but what's happening as the climate is moving out from underpopulations and ranges are forced to shift across the landscape. So how quickly can species move and adapt to find newly suitable areas? And what are the chances of evolution actually rescuing them so that they can stay in place in areas that become increasingly inhospitable? And so to tackle these questions, we work in a variety of systems. So near and dear to my heart is the clade of plants known as the monkey flowers that are centered in California, but we actually do some work up into Oregon as well. And here we do sort of field observations and experiments across species geographic distributions. I've recently started working with mesocosms in the lab, where we can create thermal gradients that simulate a very linear and narrow range and let these duckweeds sort of expand and evolve their ranges over the matter of days and weeks in real time in the greenhouse. And I've also become involved in work in the North Cascades in Washington and British Columbia, resurveying historical vegetation plots to detect recent community change and the ways that species are shifting their distributions and the way that community assemblages are changing as different species move in and out along elevation gradients. So I say all this because I want to emphasize that I am a field biologist and we work in these empirical systems, conducting our own primary studies. But I'm not going to talk to you about any of this today. Instead, what I'm going to talk to you about is something that we took a detour into a few years back, a data synthesis project that turned out to be incredibly fortuitous, given all the sort of curtailments of research that we underwent during COVID, when we weren't able to be in the field. But it was actually prompted earlier than that by some field-based results that we were gathering a few years ago. And so the first thing that prompted the study that I'm going to talk to you about today were some results that Chris Muir brought in after his field season in California. So at the time, Chris was a postdoc working with me at UBC, and he was studying the scarlet monkey flower, and he was interested in questions about the evolution of their physiological tolerances and how sort of niches were labile or constrained by different evolutionary factors. And so the details don't matter, but he had done this really large scale field experiment where he'd collected populations of the focal species from across its range and then planted them into two common gardens. So one down in the southern portion of its range in San Diego County, and then another up in the northern Sierras, not too far from where the Dixie Fire burned this summer, actually. And so in doing this study, one of the very first things we wanted to examine was the strength of local adaptation, or how well the southern populations would do in the south and how well the northern populations would do in the north, to understand how they'd evolve differences to fit these very different environments and habitats. And so his data that he brought in after the field season looked something like this, where we were looking at are estimates of the fitness of different populations to that point for different populations. And so here, each population that he planted in the garden is going to be a point on the x-axis, and they're going to be arrayed from south to north. And so I've put a star to indicate which is the local population in that garden. So the southern population in the south and a northern population in the north. And our expectation, if these things are locally adapted, is that the starred population should have the highest fitness in each panel, and who it is should switch based on the garden. And so looking at the data from the southern garden, everything looks as expected, a sort of classic example of what looks like local adaptation, where the highest fitness is for the population at home. And as you come from increasingly far away, your fitness declines to lower and lower levels. But when we looked at the data from the northern garden, we actually see the same basic rank order, which is that the southern populations are still doing the best, even though now they're very far away from home. And in fact, they're outperforming the northern populations in the northern population's home site. This is a little odd, but we talked ourselves out of being worried because, you know, these are long-lived perennial plants. This is only the first year or two of data. And so we just expected that over time, these northern populations would catch up and start to exhibit whatever traits would give them a home site advantage and let their fitness sort of become higher than the southern populations with just a little more data. We just needed more time for the data to sort themselves out. But literally the same week, I had another meeting with another student in my lab. And in this case, it was Megan Bontrager, who was doing her PhD at the time. And so Megan was working on a totally different species, a different family of plants. This is the Clarkia pulchella that grows in a different biome. So here it's in sort of the drier portions of Eastern Washington and Northeastern Oregon into Idaho and Montana. And so she had collected populations from throughout the northern half of its range, and then propagated them into 2 common gardens at the very northern edge of its range to understand the dynamics at that northern range edge. And so she brought in her field data where she'd measured lifetime fitness, because this is a little annual plant. And so this is lifetime seed production. It's a very good proxy of its total fitness. And when we look at all these populations and look at whether they were from the local common garden sites or from these populations from throughout the northern half of their range, there's tremendous variation, but no local advantage on average. So again, foreign populations, many of them were actually superior to the local populations in this Clarkia system. And here we didn't have to wait any more time. There was no hope of them recovering fitness advantage over time, because this was it. They were dead. They're just annual plants. And so sort of started scratching our heads, because, you know, we're raised on stories of local adaptation, right? Local adaptation is supposed to be ubiquitous, right? And you can probably think of your favorite examples of color matching in animals, or sort of these classic elevation gradients where plants adapt in height and other traits to the alpine environments, or whatever the trait is. We know that local adaptation gives us these like intriguing and compelling examples of all the complex ways that populations evolve to fit their natural environments. We think that local adaptation is often an engine for diversification, sometimes actually leads to speciation. And despite all of these examples, for whatever reason, my lab was unable to detect it in a couple of different studies coming in sort of really close in time. And so we started to think, well, what is it? Are we just not doing it right? Are we not doing them, you know, using appropriate methods, or is something else going on? And so we started to dig into the literature and look for compilations about the ubiquity of local adaptation, and recalled this very nice meta-analysis that Joe Herford did quite a number of years now. And so he was interested in quantifying the strength and prevalence of local adaptation from all of the studies he could gather. And what we're looking at in this summary figure is a sort of bivariate space of your fitness advantage when you're the local population compared to your fitness advantage when you're elsewhere, when you're the foreign population. And so if you're locally adapted at home, but the local population is locally adapted when you're foreign, then local adaptation should appear in this other quadrant, right? But it turns out, and that's about 75% of the data points are in this local adaptation quadrant. But it turns out there's actually quite a bit of data that suggests that populations aren't locally adapted. They might be the best population no matter where they go, They might be the worst population no matter where they go, even at home, right? Or you might get these weird reversals where everybody does better elsewhere, right? And so in a nutshell, what I want to explore in this talk is sort of why are so many populations actually ending up in these seemingly maladapted quadrants, right? And what explains this variation? And when can we predict that we should see local adaptation? And when will we see that local adaptation actually fails? Okay, and so what I'm really, the big biological question here is, what explains this variation? So I'm going to structure the talk, first of all, by telling you about the database that we assembled to try to tackle this question. And then we're going to use this database of field transplants that assess local adaptation to try to explain the variation in the database. And we're going to look at that variation in a few different ways. Firstly, over time. Secondly, across space. And what I won't talk about today, because there's not time, is a third effort that we put together, which is to look at how species interactions affect the strength of local adaptation. So if anyone's interested in that topic, please just get in touch with me and I can tell you more about that one. But first about the database. So let me tell you about the kind of data that we're going to be using to tackle the study. And so this was a huge group effort. It was spearheaded by Megan, who was, like I said, a PhD student at the time, essentially did another PhD's worth of work on this sort of what was supposed to be a quick and easy side project. Of course it wasn't. But so she organized a whole team of folks to search the literature, extract the data, and then collate them in a way that we could use. So we did a formal Web of Science search. We, based on our search terms, it returned many thousands of studies that we had to screen and then attempt data extraction for a little over 200 that met all of our criteria. And in the end, by the time we were able to extract the data, contact authors, get everything we needed, we have about 150 studies and a little more, a few more than that in terms of study species that were usable for the analysis. Now, we wanted this to be a global meta-analysis. We were agnostic with regard to geography and taxonomic group when we searched, but here, warts and all, are some of the biases that were unavoidable in the data set. So first of all, people really aren't in the business of transplanting elephants, it turns out, and so it's functionally biased to things that are readily movable and trackable, and so of course it's biased towards plants, short-lived plants. Annual and perennial herbs for the most part. It is also spatially biased. So we are, of course, over-studying or have overrepresentation of the Northern Hemisphere, very few studies from the Southern Hemisphere. And so all of our inferences are really sort of limited to, despite the scale of the numbers of populations and the numbers of species, our inferences are really limited to plants of the Northern Hemisphere. And in terms of fitness estimates, about half of the studies got something like a lifetime composite of fitness that incorporated both survival and reproduction. Other studies had only one or the other of those. And so again, we've accounted for this variation, but just to point out that even though we're trying to assess fitness in the natural environment, in many cases we're falling short and getting only a piece of the organism's fitness, particularly for the longer lived species. Okay, so those are the studies we're going to use. And now I want to talk to you about how and why we were interested in exploring variation in local adaptation over time. And to set this up, I want to go back to that Clarkia example, that one of the ones that prompted this study in the first place. And so recall that we looked at the lifetime fitness of these Clarkia populations, whether they were foreign, coming from elsewhere in the range, or whether they were local, coming to these two focal gardens, the F1 and F2 sites here at the northern range edge, and found no advantage of being from the local population on average. One thing to keep in mind, and one thing that we began to explore, was that the experiment conditions when the study was conducted in 2016 and 2017 were anomalously hot. And so here's temperature, a growing season temperature on the x-axis. And the colored dots show you the historical climate conditions of the populations where they were sourced. And so here's the historical expectation for temperature, and on the y-axis precipitation for the two common garden sites, the F1 and F2 site. But when Megan put these seeds out and followed the fates of these plants, it was actually quite a bit hotter than that historical average. In fact, it was so hot that it was more like the temperature of several southern populations that were collected way down here when she did the experiment. So when we consider the temperature of origin, the historical temperatures to which these populations were likely adapted, things start to make a lot more sense. So the local populations here have this black bar for their average performance. The foreign populations that come from somewhere historically warmer were the ones that were doing really well, and the populations that came from cooler places actually did the worst of all. Okay, so this is a signal perhaps that climate change is starting to undermine the local advantage that these populations might have had in their home site. And so in the context of this Hereford study, our hypothesis became that climate change is actually overturning what was historical local advantage and causing populations to be either better adapted to sites that are not at home. Perhaps these warm populations are going to be superior across many, many sites, whereas cool populations from historically cool places will become inferior across most sites. And so this is what we wanted to explore with our transplant database. And so we're essentially asking, first of all, how robust is fitness to variation in climate in the first place? And is local adaptation, or sort of the relative fitness of different populations, is that a fitness advantage from being local actually becoming weaker or even going away as climate change progresses and climate anomalies become more and more severe? So I've told you that we did a Web of Science search to extract, find transplant studies and extract their fitness data. And what we're going to use as response variables from all of these studies are the relative fitness of different populations in the different sites, and then local adaptation, which I'll tell you about in just a moment. My dog's going to help out. And I want to give you just a concrete example from one particular study. This is one of the 160 or so species that is in the database. It's white bark pine. And so a typical study design looks kind of like this, where you have populations that were collected, shown as the colored dots. So in this case, there's 7 populations that they were working with, and a full reciprocal design where you would plant 7 populations fully reciprocally among all 7 sites is actually pretty rare. And so we often have something like this where there's 2 common gardens shown at the rectangle. So this is a southern population, a southern garden and a northern garden, but within each garden you have all 7 populations planted with replication. And so you would track these individuals and measure things like survival and growth, and probably not in this species, but maybe in others, their seed output as a measure of their relative fitness. And what we want to do is actually come up with a metric of the strength of local adaptation. And one really good component of local adaptation, a really robust way to look at it, is to think about the local versus foreign population's fitness. And so we're going to use this log ratio of local and foreign relative fitnesses as our metric of local adaptation. And for, say, the southern garden in red, we're going to be comparing the performance of each of the other source populations to the local red population in this red site. Okay, and so anytime the local population does better, this metric will be high. When the foreign population does better, this metric will be low. Okay, so that's sort of one aspect of what we need to test this hypothesis about climate change, taking away local adaptation. The other thing we need are climate data. And so for each of the sources and sites in the transplant database, we extracted the lat-long coordinates of the populations and the gardens. And from those, we extracted yearly climate records and used those to calculate the temperature and precipitation anomalies that the populations were experiencing. And so the temperature and precip data that we used were downscaled, I believe, in North America, at least from PRISM, and based on the climate NA algorithms. So this is a downscaling algorithm developed at UBC initially for Western North America, where the incredibly topographically complex landscapes make coarse gridded data pretty inaccurate. And so it's essentially trying to infer based on adiabatic lapse rates what the local climate might be even in a mountainous area. And so theirs are now available worldwide, and that was the source of our climate data. Just gonna ask my husband to please take the dog away. So hopefully, I don't know if you guys can hear her whining, but she's scratching at my door. Sorry about that. Okay, so with the climate data in hand, what we wanted to do is calculate what we call a climate deviation. This is a metric of an anomaly relative to historical averages. So we defined as our historical reference period the decades from 1950 to 1980. You know, prior to this, the records are really just too sparse. And after this, the signal of warming becomes pretty dramatic. And so we are going to use this sort of 30-year window to set a baseline to which we would define as our historical average, and then calculate the deviation as whatever the conditions were during the experimental transplants minus that historical baseline average. And so in thinking about how this is going to affect fitness, our first supposition or sort of assumption is that interannual climate variation within a site should affect mean fitness. And so if you think about, say, this population, this blue population planted at home, you know, the blue site has some historic temperature average, but with some variation around that from year to year. And so, you know, if it's adapted to the long-term average, we would expect it to do best in a year that matches the average, and a year that's a little colder or a little hotter might lead to some moderate fitness declines. And now, if we want to know how robust fitness is to these kinds of variations, we could wait a very long time and try to get some extreme years. But the handy thing about these transplants is that they experimentally expand sort of the range of climate deviations that we can observe fitness under. So if we take this blue population and plant it at a red site that has a much higher historical average, now we can observe its performance in these extreme conditions that would be pretty hard to wait for, or require a long time to wait in the blue site. Okay, so with these sorts of data, we can ask sort of how broad or peak are these fitness curves as populations respond to climate deviations. And when we think about local adaptation, then we have to think about what is the historical average of each population relative to the experimental conditions they're experiencing. So, for example, if we were to do a common garden at the blue site under conditions that are typical of the blue site, then we would expect these blue individuals, this blue source population would have a strong local advantage, right? But if we were to do the transplant in a very hot year, an unusually hot year for the blue site, these red populations planted in might have actually a substantial fitness advantage. And so we would see the opposite of local adaptation. We would see a foreign advantage. Okay, so this is sort of the conceptual framework we're using. What we do is we take the fitness data from these transplant studies and we model what these Gaussian curves for fitness as a function of environment, where we have relative fitness as a response variable. And then this sort of Gaussian equation describes the maximum fitness, which can differ among populations, as a function of this climatic deviation. So we'll express this as either the temperature or the precipitation deviation between the experiment conditions and the historical average. And then this is sort of the strength of selection for deviating from the optimum. And this is basically the rate of fitness line as you move away from the peak of the curve. And this is a parameter that we can allow to vary in our modeling as well. So I won't delve too much more into the analyses, and I'm going to cut straight to the results, and I'm happy to take questions about this for anyone who's interested. So looking at the data, so this, first of all, how does fitness respond to temperature deviation? So we've got fitness on the y-axis, and we've got that temperature deviation, or sort of the mismatch between the experimental conditions and what a population's long-term average was, okay? And so the populations that were planted at home are shown light gray. And you can see the sort of slim range of what is essentially just interannual variation in temperature that a home population can experience at home. And then the dark points, the black points, are all of the populations that are moved far away from home. And you can see how that dramatically expands the range of deviation that we can examine their fitness responses to. And then we're going to fit a separate curve for each kind of data, whether you're at home or whether you're foreign. And so you can see that the fitted curves are unimodal. So there is an optimum temperature at which fitness peaks. And when you move away from that temperature, then fitness starts to decline. So, and the difference between the gray and dark gray curves here is a home site advantage that we can't attribute to climate. We can't attribute this to temperature or precipitation. It's all the other things about that being local, whether it's soil or competitors or pathogens or whatever it is, that give you a home site advantage once we've held climate constant. Okay, but nonetheless, as the temperature mismatch increases between what you're experiencing and what you historically evolved to, your fitness does decline. But you can see that these curves are not really, really narrow, right? So it takes, this is going up to 10 degrees Celsius of temperature mismatch before you've started to fall a fair ways down this curve. Now, when we look at precipitation, the patterns are completely different. So there isn't a unimodal response. The slopes here are quite weak, but if anything, it seems that wetter is better. If you move any population to a somewhat wetter site, then they tend to have a slight fitness increase. And that's true whether you're at home or whether you're away. But these slopes are really not that meaningful in the first place. And so for the most part, I'm going to focus on temperature for the rest of the analyses here. So going back to, so this is how fitness varies with climatic deviations, but the question is, what about relative fitness rankings? So what's happening to your home site advantage or your foreign advantage as climate deviations progress? So it's easy to think, sort of, we started by thinking about only the situation where local adaptation would go away. Say in an anomalously hot year, this cooler population would lose its home site advantage and the red population would do better. But actually it could just as easily go the other way. So if the climate deviation that year is towards cooler conditions, then it might actually reinforce or even enhance the strength of local adaptation for this cool population relative to warm population in a particularly cool year. So when we split our data into whether the foreign populations and local populations were experiencing reinforcing temperature anomalies or counteracting temperature anomalies, we can explain a lot of the variation in the strength of local adaptation. So we've got temperature anomaly on the x-axis and our metric of local adaptation on the y-axis. And so if we have an increasingly anomalous temperature environment, and that anomaly is going in the direction of the home site's average, right? So a warm population at home in a hot year, then local adaptation tends to be stronger and stronger. But if we have a temperature anomaly that's going against that home site advantage, so say a cool population in a hot year home, then its local adaptation is going to decline until we can actually see this curve dipping below zero, below the break-even point into foreign advantage under extreme anomalies. And we know that actually these thermal environment, that climate change is happening, things are becoming weirder and more intense. We're seeing all sorts of climate extremes. And of course, that's not something I need to convince you of, but I want to show you that it's actually true in these data. So these are the year of study from the studies in the data set. This is the temperature anomaly that we were looking at. And studies that were done in the early years of the data collection were sort of near long-term averages, or at least no tendency to be warm. Whereas the studies in the last decades are, well above this line for the historical average, showing increasingly warm anomalies, increasingly unusually hot conditions under which we're doing these transplant studies. There is no trend in the precipitation data, whether we look at them in an absolute or a relative sense. So we know that local adaptation, or the strength of local adaptation, is sensitive to the amount of anomaly. The anomalies are increasing. If we put it together, what does that mean? So one thing we can do is just sort of divide the data into studies where the conditions were similar to the historical average, or conditions actually quite a bit warmer than the historical average. And so if we look at those particularly early studies where the studies were being done under historical similar conditions, then all local populations had an advantage, whether they're being compared to a warmer source or a cooler source, okay? But if we look at the studies that were done under these sort of dramatically warmer conditions, then the local population still does better, than cooler populations, right? But its advantage over populations that come from warmer places has all but gone away, right? And so now we see that the foreign populations and the local populations are really on par. And whereas under historic conditions, they would have had an advantage. Another way we can think about this is by looking at what's the probability that we detect local adaptation as a function of the degree of temperature anomaly. So in the top, we're looking at the probability that the local population is the best one in the set at a given garden. And that probability just goes down and down and down with temperature anomalies. The other way we can look at it is what's the probability that you do best at home compared to somewhere else where you were planting or where you were planted. And again, the probability that you'll do better away from home is increasing, and the probability that home is best for you is just going down and down as these temperature anomalies get bigger and bigger. So to try to put it together, at least for this part, the sort of glass half full finding is that although we see these unimodal fitness responses to temperature deviations, it takes a pretty large swing to start to see a big fitness decrement. And so fitness does seem actually remarkably robust to really large temperature swings. Nonetheless, as these anomalies increase, we do see a signal that local adaptation is just becoming less prevalent as climate deviations get larger and larger. And so what I think this means altogether is that we have the potential for really, really strong selection, but we don't know what the demographic consequences are. So when a population is no longer the most fit at its site, and when relative fitness is dropping a little bit, are these populations still stable, or are these fitness decrements enough to actually cause population declines and need to bring in genotypes that are better adapted to the conditions? So that was sort of what I wanted to say about how local adaptation seems to be changing over time. I want to shift gears a little bit and talk about another way that we examine these data, which was to think about how local adaptation might vary across space. And this is sort of getting back towards some of the original questions that I posed about why do populations have geographic range limits and how does adaptation change as you move towards range edges? So again, we're just trying to ask, so maybe some of this variation in local adaptation can be explained through climate change, but maybe there's another amount of this variation that can be explained by thinking about where populations are and where the gardens are within species geographic distributions. And there's quite a bit of theory that's been developed around this topic, and I don't want to go too far into it, but I just want to sort of pose a few different hypotheses that are out there in the literature. So one hypothesis would be that edge populations are less locally adapted, and that is because they might lack appropriate genetic variation to evolve adaptations to marginal environments. And so the idea here is that there's essentially a swamping gene flow that along environmental gradients that causes populations to be maladapted. As they're maladapted, because of this sort of mismatch between the influx of immigrants and the environment, the populations become much smaller, so they undergo genetic drift, so they lose genetic variations, they become maladapted, and you sort of hit this expansion threshold where populations are so mismatched in their phenotypes to what the environment demands of them that they can no longer expand and a range limit arises. So that's one hypothesis, thinking about sort of equilibrial range limits that are held in place by a failure to adapt to the environment. There's another set of theory that's looking not at equilibrial limits, but at shifting ranges and thinking about these populations that are at range edges that have just gotten there. So they're younger, perhaps they've had less time to evolve adaptations that would make them highly locally adapted to their environments. And there are things about the expansion process itself that involves sequential founder events, essentially repeated bottlenecking of genetic variation that might allow even maladapted or deleterious alleles to be carried and rise to high frequency just by chance. This is called expansion load, and it would cause populations to be harboring a lot of deleterious alleles that would lower their fitness and perhaps reduce their ability to locally adapt to their environments. On the other hand, there are lots of ideas out there that perhaps edge populations are most locally adapted, and that's because maybe they're in extreme marginal environments. At the sort of climatic or other extremes of the species range, they might have experienced strong divergent selection to evolve unique adaptations to these marginal environments. They often are fragmented, and because of that, they are free of the maladaptive effects of swamping gene flow, perhaps, and so they can diverge and evolve local adaptations. And particularly at sort of trailing ranges or ranges, areas of the range that might have been refugial during past climatic fluctuations, we might see old populations that have been there longer and had more time to evolve adaptations to the local environment. So we've actually got a lot of sort of competing hypotheses in the literature for how local adaptation is expected to vary over space within a species range. And so, edges that are limited by drift and maladaptation should have low local adaptation with a peak at the range center. These hypotheses about expanding range edges really just focus on what happens from the core to the leading edge of a range. And whereas populations that are in sort of range edges that are extreme or isolated are expected to have high local adaptation, those could be sort of just at the trailing edge or at any edge, really. These are sometimes agnostic to which kind of edge you're at. And along with these predicted patterns of difference in local adaptation, we also have expectations about population quality, where these edge populations might harbor a lot of genetic load in these hypotheses about drift and maladaptation. They might harbor load because of expansion dynamics under the expansion hypothesis, and they might have, in these extreme and isolated environments, might have gone through inbreeding or other sort of consequences of small population size that would again reduce population quality. So what we wanted to do then was to sort of test for these patterns in the transplant database. And so I've already told you about this sort of pathway all the way through local adaptation. I want to mention how we estimated population quality from the same data. And so I'll go back to that white bark pine example. And again, remind you that these are sort of source populations as dots planted into these two gardens. I'm cartooning with the rectangles where we're looking at the fitness of each source in each of these two common gardens. And so population quality is essentially the average performance of a population across however many sites it was planted into. The only thing that makes this look a bit complicated is that we first have to normalize the fitness of any given population to the average performance of all populations in a given site, because sites differ in productivity, differ in, say, resource availability. And so we want to normalize performance to those site level differences before we calculate a population's average performance. And so, for example, for the red population, its population quality would simply be its normalized average fitness between the blue and the red gardens. Okay. So then the other piece we need to do this then is a description of species ranges, so we can quantify sort of where a source and a site are within a species range. Are they at the core, are they at the leading edge, or say a trailing edge? And so for that, we relied on GBIF records of occurrence, mostly museum and herbarium records, which have a whole bunch of caveats that I'm happy to talk about with anyone who's interested. But suffice it to say, we did the very best we could and compared the GBIF records to atlases to make sure that they were faithfully representing at least the outline species range, and used them to map distributions. This was a whole other data collection effort that was again led by Megan, and this time involved a number of other students and postdocs in my lab at UBC. So let me just walk you through another example. This is still the white bark pine, and this time you're looking at its occurrence records from GBIF as the tiny dots. And we just simply described its range by putting the minimum convex polygon around those points. We tried all sorts of other fancy things, but in the end, we just used the MCP. And the range center would be the sort of geometric centroid of this polygon. And so what we need to derive from these distributions then are predictor variables about geographic and climatic range position. And so, for example, we can think about what is the relative latitude of any given source population And so here we just simply looked at the min and the max latitude across all the records of occurrence and characterized each site sort of within that span. A low value would be at its sort of equatorial boundary, a high value would be at its polar boundary, and a value of 0.5 would be right near its range center. And then, so this is using latitude, really thinking about that expansion axis for Northern Hemisphere species, proceeding largely from south to north. But we also had a metric that was sort of regardless of direction, the directionless metric of centrality, which is basically the ratio of the distance of a given source or transplant site to the edge of the polygon relative to the centroid of the polygon. Now for climatic range position, because a lot of these hypotheses invoke sort of selection of marginal habitats, and one of the primary ways we can think about environmental marginality is to think in terms of climatic marginality. And so what we did was we extracted for each of the occurrence records, the mean annual temperature and mean annual precipitation, and then use that to create sort of a histogram of the climatic conditions that are experienced across the species range, and then characterized each source and transplant site by where it fell within the climatic range, similarly to how we dealt with latitude. But in this case, taking account of frequency of records and not just the maxes and the mins. Okay, so apologies for this slide. All I want to say here is that we are using our response variables of local adaptation or population quality, and modeling them as a function of our geographic range position variables, our climatic range position variables, and some covariates like where actually was the site in absolute space, its absolute latitude, and how far apart are the populations in the study, because we would expect local adaptation to increase as the difference between sources and sites increases, as well as a bunch of random effects to account for the different levels of variation in our data set. Okay, so all by way of saying what we're trying to do is answer these very basic questions. Is local adaptation stronger or weaker at range edges? And is population quality lower at range edges? And how does this help us understand variation and adaptation? Okay, so let's look at the first question. How does local adaptation vary with range position? So here we've got local adaptation on our y-axis in both panels. The left-hand panel, we're looking at that metric of relative latitude, whether you're at your equatorial edge, at your range center, or your polar edge. And in the right, it's a directionless estimate of centrality. So are you at the periphery or are you at the core of the range? And what we find when we actually put all the data into this analysis is that local adaptation tends to decline towards polar edges. So there's strong support for a negative coefficient, a linear coefficient, meaning that we have sort of a decline as you move from the equatorial edge through the range center towards the polar edge. We don't find any support for local adaptation varying with this metric of centrality. It's only when we think about the latitudinal acts of geographic marginality that we detect a decline in local adaptation. We can do a similar sort of investigation for climatic range position. And so here, instead of the geographic marginality, we've got thermal marginality on the left and precipitation marginality on the right. So are you cold, central, or warm? Are you dry, central, or wet as you move across these x-axes, but still local adaptation on the y-axis? And what we find is that local adaptation is actually stronger in thermally extreme sites, whether they're cold or warm. And we don't find support for any varying local adaptation with regard to precipitation. And I want to point out that although a lot of the theory assumes that range edge sites are climatically marginal, we find that climatically marginal sites exist range-wide and not necessarily with any greater prevalence at the range edge, which I think is pretty interesting. So here we're just color coding the records by degree of thermal marginality, from blue being cold to red being warm. And then we're arraying them with regard to latitudinal position. So are you at the equatorial edge or the polar edge or right in the range center? And you can see at any given range position, there's warm and cool sites available. And at the range center, you could actually be quite cold or quite hot relative to the rest of the species range. Okay, so let's look at population quality. So now this is the same setup as the initial graph, those earlier graphs. We've got our latitudinal range position on the left-hand panel and our thermal range position on the right-hand panel. But now instead of local adaptation, we're looking at population quality on the y-axis. And so we see that actually population quality does seem to decline towards both kinds of range edges with a peak in the middle of the range. And it tends to be higher in cool sites and lower in warm marginal sites. So populations that come from latitudinal range edges and warm edges tend to be of lower quality. So what does this mean when we put it all together? So if we think about those polar edges, then these populations seem to be poor on average. So they're of low population quality, and they're poorly locally adapted. So they're not doing well at home, and they're not doing well on average anywhere else either. And so this does suggest that they might harbor some sort of genetic load. This is consistent with the expansion hypothesis, but of course we can't say for sure that's the driver. Now, at the other end, at sort of equatorial edges and warm edges, we see that they're both, they're poor on average, so they have low population quality across most sites, but they do really well at home. And so this suggests that they're highly specialized, which is consistent with those hypotheses about being isolated or having a lot of time to evolve highly specialized local adaptations that might just incur cost in other environments that aren't at those kinds of edges. And if we think about sort of all of these results in sort of put together, what I hope I've convinced you is that these kinds of syntheses can start to help us explain sort of when and where local adaptation breaks down, give us some reasons why these sort of dots out here and these other three quadrants that don't show local adaptation aren't just mistakes, but they actually have some we can explain based on the way that climate change is pulling the rug out from under populations, or the way that expansion dynamics cause some kinds of populations to harbor genetic load, why we fail to see local adaptation in all cases, despite strong selection. And zooming out even just one step further, when I speak to ecologists and evolutionary biologists, I don't often, I think, have to explain why local adaptation matters, but just to be really clear, I mean, the genetic variation within species is a key component of biodiversity. It's cryptic. We don't often see phenotypic markers of local adaptation that we know have genetic bases. But nonetheless, the way that these sort of physiologies and other traits differ among populations within a species range is a key element of biodiversity, and understanding its drivers and why it breaks down, I think, is a really fundamental problem. But it does also have some implications for how we conserve and manage populations. So for example, the mantra for restoration ecology has been local is best, right? And when you're selecting populations or seed sources, then you would want to use from within a seed zone, or as close as possible to the restoration site. And the fact that these local populations are just losing their home site advantage, and all of those aspects of the local biology, be it competitors or pathogens or predators, they seem to be sort of fading in importance as climate anomalies get bigger and bigger, I think causes us to have to question, what is the role of finding pre-adapted genotypes elsewhere in the species range that might help populations keep a foothold in sites where they historically have been present. It also, there's been a lot of discussion in the literature over the years about whether edge populations are worth saving. Are they the next best hope for range expansions, or are they sort of full of genetic load and perhaps not worth conservation emphasis? And again, I think these results help speak to the value of maybe conserving sets of populations in a region, any one of which might harbor some maladaptive alleles, but nonetheless, the mix could preserve whatever local adaptations are there, while sort of masking some of that genetic load. So with that, I will stop, and I hope that I can take some questions. But before I do, I just want to acknowledge all the folks I mentioned as we went who have contributed so phenomenally to these massive data collection and collation efforts, but also acknowledge the fact that this is built on the backs of all the folks who've been out in the field conducting those reciprocal transplants in common gardens and making their data available in the literature, as well as Joe and Chris Eckert, both of whom were able to share their prior efforts at collating these sorts of transplant data. And thank you guys for your time and your attention.
Thank you, Amy. That was fantastic. And I really appreciate how at the end you brought it out to the big picture, talking about all of our conservation strategies. I really appreciate that. Anyone who would like to ask a question, you can put it in the Q&A or in the chat box, whichever you're more comfortable with. While we're waiting for some questions to come in on the Q&A, Amy, you talked about how you had to downscale the climate data because, especially in your part of the world, it's so mountainous and so diverse in terms of elevation. And so I wondered if you'd had a chance to look into the literature, do any experiments on elevation effects. I'm talking about latitude and temperature, but we also think about species moving, up or down in that sense.
Yeah, for sure. So yes, so the last study that I was talking about, where I was focusing on latitude, we also examined things with respect to elevation. So where were you within the elevation range of a species, and did not find very strong signals there at all. One thing I worry about, it was surprisingly hard to characterize species elevation ranges from GBIF, just because that elevation column is often blank. And so I worry that our lack of signal is not biological, but more just sort of data quality on that front. But certainly that's an important axis to think about. And I like to think about whether they're substitutable. Do we fail to see these big northward shifts because things are just like scooting uphill or around the mountain? And anyway, I don't have much to say concretely about that, but I do think it's an important angle to bring up. Thanks.
Rich area for future projects.
Yes.
All right, so there's a question in the Q&A from David Lytle. David, do you want, Teresa can turn you on so you can ask it out loud if you'd like.
Hi, David. Sorry, I'm just chiming in. If you can raise your hand, then I can allow you to speak. Perfect, thank you. Okay, now if you just unmute, you should be good to go.
Hey there, can you hear me?
Yes.
Yes.
Okay, great. You probably can't see me though. I'm in here with my lab group. We're in lab wearing masks. Anyhow, nice talk. Thanks for presenting that. So my question is, as you're well aware, Dan Jansen had this idea back in the 1960s that when you have, in the tropics, you have such little climate variability that might select for species having very narrow thermal tolerances. And so you get a lot of species packed into these elevational gradients. And some of the findings that you have reflect that idea, but the range that you're looking at doesn't really go all the way to the tropics. So I'm wondering, do you think any kind of process like what Jansen invoked might be operating underneath some of the patterns that you've seen here, or is it something else entirely?
Yeah, that's interesting. I guess I haven't thought about interpreting the latter study's results in light of that Jansen hypothesis. And I would be a little skittish to do so, just because most of our studies, although we're talking about equatorial edges, that's relative, and they're still temperate species. Very few things are truly equatorial. Thought about it a little bit more in the context of the climate change study, because we had that sort of variance parameter, sort of the way that the penalty for falling off your optimum, and we let that parameter vary. And so I would like to, that was something we didn't really focus on interpreting or analyzing, but I would like to go back and see, is that a parameter that varies predictably, say with latitude, in a way that I think we could say would be consistent with the Jansen hypothesis. But again, mostly temperate species, so I'd be a little bit skittish to make too much of it, since we don't really capture that true tropical axis.
Right, it's just the similarity is compelling, so I can't help but wonder, but,
Yeah,
okay, thanks.
thanks for the question.
Thanks. All right, Bob Mason.
Can you hear me?
Yes.
Yes.
Okay, good. I can't see my unmute here. I'm like Dave, actually, sorry, let me move my window off of Amy's face. There you go. I like the talk too, it was great. My question is, where you live in Western Canada, but perhaps further to the north, is within a belt that is experiencing some of the greatest temperature changes due to climate change. And of course, same thing on the Southern Hemisphere as well. Is this resulting in an unfortunately fortuitous greater degree of change that you can be, that you're observing because of where you're doing the common garden experiments, as opposed to say you were in, I don't know, Kansas or something, or much further to the south? Is that something you've ever thought about as well? If that makes sense.
So yeah, I do think about how that that perturbation varies spatially for sure. I do want to clarify though, these although I started the talk with a couple of like vignettes from my lab, the results I was presenting were actually collated from across the published literature. So they come from all throughout North America and Europe, and there's some studies from Australia and South America and elsewhere, but predominantly Northern Hemisphere, but certainly broader than my local region. But one thing I also forgot to say, but I should have clarified, because we knew from the first study that these climate anomalies were having a pretty strong imprint on the patterns of local adaptation. When we did the spatial analysis, we put that climate anomaly in as a covariate. And so the spatial patterns are sort of adjusting for those temporal patterns as well. So they're not confounded, that the northern gardens are actually just experiencing the heaviest anomalies because of that spatial patterning and climate change. I should have clarified that. But yeah, that that would be a pretty big confounding factor, that if we hadn't adjusted for that.
Yeah, and I'm not trying to suggest that I wasn't even thinking that it was confounding your data. I was just wondering if that's just a this is just one of those lucky happenstances that you're not in Texas or something, and it might be a little harder to well, we could explore that for other reasons, but it might be harder to see some of those really, it looks like kind of dramatic changes that you were able to measure there. So it's pretty neat. Any rate, that wasn't a jab at your data. I think I was just kind of wondering about that. It was just like, wow, that's kind of interesting. I wonder if we're going to be limited to people either in Chile or up in Canada and whatever, I don't know, Russia or something, whatever, being able to really see these dramatic effects, unfortunately.
I like your spin. This is, we'll make lemonades out of lemons. I often feel pretty sad when I come back from a field season.
All right, but anyway, very nice. Neat stuff.
Thank you.
I had another question, Amy. You put out early on the slide how the meta-analysis was really biased to these short-lived plants. What type of organisms would you like to see more studies done in? You had a few fish, but I imagine they were probably freshwater fish. Where do you feel like would be a great direction to go for students to pick up new projects?
Well, so clearly, like, it would be really hard to do this in highly mobile, big giant creatures that require all sorts of permits to move around. So, I'm not going to propose that a viable thing is to start, transplanting cougars or something. But, I do think like we could do more with enclosures, like cage experiments, small mammals. Certainly we should see more insects. People have done transplants with butterflies and fruit flies, just not that many, at least not that we were able to uncover. So, stick with the small, relatively short lifespan things, but I don't think they need to just be plants, would be my naive botanist answer, not knowing what the foibles are. Lizards, we can catch lizards and move them around, right?
All right, Bob, where are your snakes? Can you move some of your snakes? All right.
I mean,
well,
there are, there's ethical questions about, how far can we move stuff, and can we do these really natural experiments where we get fitness, but we let these things escape? Or do we have to sort of contain the foreign genotypes in a way that truncates our ability to estimate fitness, but at least we know that we're not tainting the local populations? I mean, I've sort of thought about that a fair bit with our work, and we often end up letting them flower and then plucking the buds off, so we don't actually get seed set, but that way we know that they're not escaping. Yeah. There are issues.
I'm sorry, I'm breaking up. It's Bob Mason again. Yeah, what Sarah is saying is I work on the garter snakes up in Manitoba, Canada, that come out by the tens of thousands. And it's so interesting. And of course, I'm getting pretty gray haired. So I've been going up there for almost 40 years. And they're being really significantly impacted by climate change, in the sense that they're going into hibernation later, and they're coming out earlier. And, it's another one. People keep asking me, well, how's that going to work out? And I said, well, probably being not underground for a longer period of time is probably not going to be the worst thing for them. But then I don't know if the frogs that they eat and so forth are going to be, they're going to be available, what have you. So as you well know, it's certainly complex. But they are also heading up north. So there's actually den sites they're discovering now that are approaching the Arctic Circle. So I mean, it's just kind of crazy that reptiles are moving that far up. And that's further to the west, closer to where you are, because of the Canadian shield, the granitic shield. So as long as they have limestone under them, they can actually move for it. But they are moving north. So it's kind of weird. But I applaud what you're saying too. I think you're right. Trying to move cougars or whatever, I like to forget that. But there are a lot of critters there, and leopard frogs. I think you said amphibians. Leopard frogs are another one that seem to be moving around a lot. There's some really good data up in Canada there, but it's extremely sporadic and so forth. But I think there probably are some good critter examples there, and I'd have to really think about it. But yeah, it's worth thinking of, and it's not pie in the sky, at least in my opinion.
All right, well, thank you. We are officially at the end of our time for the seminar, but like I said, I'll send an email with Dr. Angert's available times for this week. And so if anyone wants to follow up with some additional questions or chit chat or study ideas, she's available. So thank you for your time today, Amy.
Wonderful. Thank you very much. Bye, everyone.
Bye.